Okay, so the topic of this video is deoxyribonucleic acid, or better known as DNA. So let's go ahead and get started talking about DNA. Okay, so what is DNA? Well, here's a fairly simplistic definition right here. It's a molecule that stores genetic information. You're often going to hear DNA compared to a blueprint. And in the picture, here's a blueprint. You can kind of tell it's a blueprint to build a boat. A blueprint is really simply a set of building instructions. And so that makes a blueprint a wonderful analogy for what DNA is. DNA is a blueprint. And what we're going to show throughout this video and others is that DNA are blueprints, or is a blueprint that tells cells how to build proteins. And we'll talk more about that in a moment. If you recall from earlier in the school year, we talked about monomers and polymers when we were discussing organic molecules. And we mentioned that there's 4 categories of organic molecules. Nucleic acids is one, carbohydrates is a second, lipids is a third, and proteins is a fourth. And all 4 kinds of organic molecules are made from small monomers, small basic units. And so the basic unit, or the monomer, of nucleic acids are what we call nucleotides. And here's a picture of a nucleotide. You can see a nucleotide is made from 3 subparts. You have a sugar group, you have a phosphate group, and then you have what's called the nitrogen base. We're going to again talk more about nucleotides in a few moments, but here's just a refresher. If we remind ourselves of what nucleic acids are, again, they are millions and millions of nucleotides bonded in a big chain. DNA, for instance, is bonded in a double chain. There's a left side to the DNA molecule, there's a right side. And so in this little simplistic picture here, we see that all these A's and T's and C's and G's, these are parts of nucleotides, and they're all bonded to make one big spiral, twisted spiral molecule called DNA. And we're going to talk more about these A's and T's and C's and G's in a moment. So if we kind of take a moment and look at this picture, here's a picture of DNA, and from the picture, how many nucleotides do you see in this picture? Pause the video if you want 30 seconds or so to look it over. I'm going to go over the answer in 3, 2, 1. So how many nucleotides are in this picture? Well, there's one nucleotide, a phosphate, a sugar, and then a nitrogen base labeled A. Well, here's a second nucleotide, a third, a fourth, a fifth, a sixth nucleotide. Every nucleotide has a phosphate, a sugar, and one of the 4 nitrogen bases in it, either the nitrogen base A, T, C, or G, adenine, thymine, cytosine, or guanine. So you see there's 6 nucleotides on the left of this picture. Well, there's a 7th, there's an 8th, there's a 9th, 10, 11, 12. There's 6 nucleotides on the left, 6 nucleotides on the right. So there's a total of 12 nucleotides in this picture. Every nucleotide consisting of a phosphate, a sugar, and one of 4 different kinds of nitrogen bases. So if we kind of come back to these notes really quick and we look at the same picture that we see here, what we actually see, these A's and T's and C's and G's that we can read, this is actually symbolic of a gene. A gene is a small piece of DNA, and what it does is it holds instructions to build a protein. Earlier I mentioned that DNA is often compared to a blueprint. A blueprint is a set of instructions to build something. So for instance, in the picture, I just highlighted, for instance, this gene. Pretend this is a gene. This is a set of instructions here to make a protein called hemoglobin. You might know hemoglobin is the protein found in red blood cells that helps to transport oxygen. So genes are small pieces of DNA, small pieces of those A's, T's, C's, and G's, and they instruct our cells on how to build a protein. And we're going to learn how they do that another day in another video. But that's kind of a sneak peek. So let's look at nucleotides in a little more depth. Nucleotides, we said earlier, are made from 3 basic parts. And in this picture, we see one of the 3 parts. We see the sugar molecule. The name of the sugar molecule of DNA goes by the name of deoxyribose. There's something in the way that deoxyribose is spelled that lets you know it's a sugar. Anytime you see molecules end with OSE, that's a giveaway that they are a sugar molecule. You think of other sugars you've heard of. You've heard of sucrose, fructose, glucose, lactose, and now in this case, deoxyribose. It's the name of the sugar that makes up DNA molecules. You're often going to see the sugar molecule, the deoxyribose, simply illustrated in diagrams as a pentagon. And that's simply because the atoms that make up deoxyribose are themselves arranged in a pentagon shape. So for simplicity, you're often going to see the deoxyribose of a DNA simply drawn as a pentagon. A second part of a nucleotide is called the phosphate group, and here's the actual atoms that make up the phosphate molecule, and notice how there's a phosphorus in the middle. Also, for simplicity, you're often going to see the phosphate groups simply drawn as a circle, maybe with a P in the middle for phosphate, but really, for simplicity, we're going to illustrate the phosphate molecules as circles. So what is unique about everybody's DNA on Earth is we all have a unique combination of nitrogen bases. And there's 4 different kinds of nitrogen bases. You might know a nitrogen base labeled A for adenine, a nitrogen base T for thymine, C for cytosine, and G for guanine. And I said a moment ago that everyone on Earth has a unique combination of A's, T's, C's, and G's. Well, that's true except for identical twins. Identical twins have the exact same combination. But everybody else on the Earth, we all have a unique blueprint, a unique combination of adenines and thymines and cytosines and guanines. It's what makes each one of us genetically unique. Well, in this picture, here are the 4 different kinds of nucleotides. And notice how they each have the sugar, the pentagon-shaped sugar. They each have the round phosphate molecules. So how are the 4 nucleotides in the picture here, how are they different from one another? How do these 4 nucleotides differ from one another? Well, it's not the sugar, it's the same. It's not the phosphate, it's the same. How they differ is by the type of nitrogen base that you see. The adenine and the thymine, the cytosine, and the guanine. Let's talk about these A's, T's, C's, and G's in more detail. So nucleotides will combine to make nucleic acids. We mentioned that a nucleotide is a monomer. So here we have the nucleotide with an A for adenine in the picture. Watch what happens. Here's one nucleotide, and here we have a bunch of nucleotides have just bonded with that original A that we saw. I now see 1, 2, 3, 4, 5. I now see 6 nucleotides bonded together. This would be on our way to making DNA, a nucleic acid. If we remember Erwin Chargaff and Chargaff's rules, and what Erwin Chargaff discovered was DNA always had an equal amount of adenine and thymine. He didn't quite know why. And he also found out that DNA molecules had an equal amount of cytosine and guanine. Again, he didn't really know why. It took years of investigation for us to figure out why adenine was always equal in amount to thymine and why cytosine was always equal in amount to guanine. The reason we now know that A's are equal to T's is because they bond and pair with one another. From the top down, if you have a cytosine, a C on the left, bonded to that on the right will be a G for guanine. So if you have a T on the left, bonded to that will be an A for adenine. The third one down will be a T. The fourth one down will be a C. The fifth one down will be a T. The sixth one down will be a G. So these are what are now known as Chargaff's rules, after Erwin Chargaff. And so what we have here is a picture of 12 nucleotides bonded together to make a nucleic acid. I have 6 nucleotides on the left and 6 nucleotides on the right. Now, if you also notice, there are what are called 6 pairs. These are called base pairs. 6 pairs of A's and T's and C's and G's in the picture. So one thing I want you to know is DNA is a really large molecule. For instance, the entire human genome, and the word genome simply means all of the DNA. So all of human DNA packed into the nucleus of every cell in our body. We've counted. There's over 3 billion pairs of A's and T's and C's and G's packed into every nucleus of every cell in our body. So DNA is a rather large molecule. In this simplistic diagram here, I have 6 base pairs, nitrogen base pairs. But in reality, DNA molecules are really large. If I just give a simpler diagram of a DNA molecule here, I've given you GATTACA. Now the reason I chose these letters is because if you've ever seen the movie Gattaca, a science fiction movie came out in the late 1990s called Gattaca. A wonderful science fiction movie for those of you who like sci-fi. I very much recommend checking it out on Netflix or something. And it's just a fun view of genetic engineering perhaps in our future. But the name Gattaca really isn't a name at all. The people that made the movie Gattaca, they just took the 4 nitrogen bases of DNA, A's, T's, C's, and G's, and they tried to arrange those 4 letters into something that resembled a title for a movie, and they came up with the name Gattaca. They easily could have done this. They easily could have named the movie Satutagita, but it doesn't roll off the tongue as well as Gattaca does. But if you remember Chargaff's rules, if you have a G, then there's going to be a C. If you have a T, there's going to be an A. Chargaff's rule. So when we look at the structure of DNA, the double helix it's called, in 1953, these two gentlemen here, James Watson on the left, Francis Crick on the right, they built a model that explained how the structure of DNA was arranged, how the atoms were arranged in the overall molecule of DNA. And what they concluded was that DNA is in the shape of a double helix, and also that adenine pairs to thymine, A pairs to T, and that cytosine pairs to guanine, C pairs to G. And before this discovery, there was some debate as to how were the nitrogen bases arranged. Were they arranged in the inside of the double helix or on the outside? And so through some research, they were able to conclude that the nitrogen bases are on the inside. Now, that research, you might know the backstory. That research kind of involves the lady here in the bottom of this picture here, Rosalind Franklin, and a fairly famous photograph that she made on top here. This is called Photo 51, and this is a picture that she made using X-ray crystallography to produce an image of a DNA molecule. And then through some fairly complex math, Watson and Crick were able to determine the structure of DNA from this picture. Sadly, there's kind of a backstory as to how Watson and Crick obtained this picture. You guys can read about that on your own if you'd like. And so what we now know about the structure of DNA and the double helix molecule is that nucleotides, remember if there's a nucleotide on the left, there'll be a T nucleotide on the right, and what holds them together is there's what are called hydrogen bonds in between the A's and the T's, and hydrogen bonds in between the C's and the G's, and that's what the flashing lines in this picture are supposed to represent. The hydrogen bonds, it's what holds the A and T together and the C and G together. And so one of the breakthroughs that James Watson and Francis Crick described in their research paper when they described the structure of DNA is they immediately saw a way in which DNA could be copied or replicated. And that's what DNA replication is. It's the process of how DNA is copied. And this takes place during the S stage of interphase. We talked about the cell cycle and interphase earlier in the school year. But during the process of DNA replication, one piece of DNA, one molecule of DNA will be copied into 2. And if you wonder why do cells make copies of DNA? Well, because cells are always multiplying. When one cell multiplies into 2 cells, both of those cells need a copy of the DNA. So there has to be a way to duplicate the DNA. And that's what I want to talk about right now in this process called DNA replication. I'm going to break it down into 3 steps. Step 1 is there's an enzyme by the name of DNA helicase. Now look how I spelled it. I underlined ASE, helicase, ASE. Anytime you see molecules with an ASE ending, that's a giveaway that the molecule is an enzyme. Earlier we saw words that end in OSE. OSE are sugar. ASE is an enzyme. So enzymes, if you recall, are types of proteins, and they help to speed up chemical reactions. They reduce the energy needed for a chemical reaction to occur. And so in my simplistic animation, a DNA helicase is represented by this animated scissors. And what the DNA helicase does is it breaks apart the bonds holding the A and the T, holding the C and the G. So one by one, DNA helicase will break apart the bonds, the hydrogen bonds between the A's and the T's, the C's and the G's. And that causes our DNA molecule to separate, to open up like you just saw in the animation. Well, what happens next? What's step 2? So step 2 is, keep in mind, this is all in the nucleus of the cell. This is where DNA is found. And the nucleus, it's not filled with empty hollow airspace. There's a fluid in the nucleus, much like the cytoplasm. And there are molecules dissolved in that fluid of the nucleus. And so our directions say free-floating nucleotides, free-floating adenines and thymines and cytosines and guanines match up. So look on the left strip of DNA. I have a C on top. Top left, I have a C. Well, I hope you a G for guanine will float on in and match up. The next one down is a T, so an A is going to float in. The next one down, I have an A on the left, so a T will float in. The next one down, here comes a C. The next one down, here comes a T. The next one down, here comes a G. Well, the same thing happens to the DNA on the right of the screen, only replication takes place in opposite directions. So in this case, we're going to start at the bottom and work our way up. There's a G on the bottom. Here comes a C. Next one up, there's a T on the right. Here comes an A. Next one up, here comes a G. Next, here's an adenine. Next, here's a thymine. Next, here's a cytosine. And so what we've created here are 2 pieces of DNA. Now we're not quite done, but that's kind of the main gist of it right there. But let's finish these up. Step 3, there's an enzyme, a different enzyme by the name of DNA polymerase that helps to reconnect the 2 strands. And I'm being a little simplistic here by illustrating DNA polymerase as a bottle of glue. And it helps to glue and bond the adenines and the thymines and the guanines and the cytosines all together. And so there's this enzyme called DNA polymerase that helps to reconnect and rebond and basically zip up the DNA and complete this process. And when the process is finished, I hope you notice both strands of DNA are identical. Look at the letter code on the top, C and G to the left, on the right, C and G. Next letters down, TNA, next letters down, TNA, next letters down, A and T, and then A and T. The DNA strands are identical to one another. Well, in the creation of all these A's and T's and C's and G's, remember there's 3 billion pairs of A's and T's and C's and G's per cell. Of course, there is the chance of a mistake being made, and we call these mistakes mutations. Hollywood's got it all wrong. If you watch a Hollywood movie, they're going to convince you that mutations allow you to shoot lasers out of your eyes and control the weather with your mind and bend metal and control metal with your thoughts. So I'm thinking of the X-Men movies. But, that's not really accurate as far as what mutations are. A mutation is a change in our DNA sequence. Notice in the animation here, I've highlighted a red letter C. C and A, they don't bond together. So that's clearly a misspelling right there. This is an example of a mutation. The good news is mutations are often corrected. We have enzymes that act as little proofreaders and spell checkers. What they'll do, I'm going to illustrate the enzyme again with my little animated scissors here. So in my animated scissors, they've found that mutation, the red letter C. And the reason I think scissors are a good analogy here is because the enzyme will cut out and remove the mutation. And once the C for cytosine has been cut out, that will allow the proper nitrogen base to fill in, thus repairing the mutation. And we've actually estimated pre-repair, before any of these mutations are fixed, it's estimated that there's about 1 error, 1 misspelling, 1 mutation in every 10,000 nucleotides. That might not seem like a lot, but remember that the human genome is made up of 3 billion pairs of A's and T's and C's and G's. If there's 1 mistake out of every 10,000, that's millions of errors right there. But after the mutations are repaired, we estimate that there's only about one mistake in every billion or so. So, there are no perfect samples. We are all flawed. Every person has 3, 4, 5, 6, 7, 8 mutations, and we just hope that those mutations are not in key locations of our DNA that could cause undue suffering. So there you go. There's a brief introduction into the world of DNA, deoxyribonucleic acid. If you're in my biology class, pause the video, try to answer these 5 questions, write your answers on a separate sheet of paper. I'd be happy to check them for accuracy. Good luck.